Module 1, Foundations of Biotechnology. Lecture 1, Revisiting the History and Concept of Biotechnology. Welcome to this course on the principles of biotechnology. Biotechnology is the use of living organisms, biological systems, or their derivatives to develop products and technologies that improve human life, agriculture, and industry. Although the term biotechnology is relatively modern, the practice is ancient. Traditional biotechnology can be traced back thousands of years to processes like bread fermentation, brewing, and cheese making. Modern biotechnology began with discoveries in microbiology, genetics, and molecular biology, leading to the ability to manipulate DNA directly. The concept evolved significantly in the 20th century, with milestones such as the discovery of the DNA double helix, the development of recombinant DNA technology, and the sequencing of the human genome. Today, biotechnology underpins fields like medicine, agriculture, and environmental management. Lecture 2, Considering Nutritional Requirements in Biotechnology. Biological systems require nutrients to grow and function. In biotechnological applications, understanding the nutritional requirements of cells is essential. Microorganisms, plants, and animal cells grown in controlled environments require balanced sources of carbon, nitrogen, minerals, and vitamins. For instance, microbial fermentation for antibiotics demands optimized nutrient media. Similarly, plant tissue culture requires specialized growth media containing sucrose, salts, and growth regulators. By controlling nutrient availability, scientists can influence growth rates, metabolic products, and developmental outcomes. Lecture 3, Introducing Morphogenesis. Morphogenesis refers to the biological processes that control the shape and structure of an organism or tissue. In biotechnology, morphogenesis is particularly important in plant tissue culture, where cells can be induced to form shoots, roots, or embryos under specific hormonal and environmental conditions. Understanding morphogenesis allows scientists to regenerate whole plants from small tissue fragments, an essential technique for crop improvement and genetic engineering. Lecture 4, Types of Tissue Culture. Tissue culture is the practice of growing cells, tissues, or organs under sterile conditions on nutrient media. The basic types include callus culture, growing undifferentiated plant cells. Organ culture, maintaining whole organs or parts of organs. Cell suspension culture, growing plant cells in liquid medium. Embryo culture, rescuing immature embryos for hybrid development. These techniques are widely used in micropropagation, conservation, and genetic modification of plants. Lecture 5, Allied Tissue Culture Techniques. Beyond basic tissue culture, biotechnology uses allied techniques such as protoplast culture, cells without cell walls, anther and pollen culture, to produce haploid plants, and somatic hybridization, fusion of protoplasts from different species. These methods expand the possibilities of crop breeding, allowing combinations of traits that are not possible through conventional crossing. Lecture 6, Structure of Nucleic Acids. The central molecules of biotechnology are nucleic acids, DNA and RNA. DNA is a double helix composed of nucleotides, each containing a sugar, a phosphate group, and a nitrogenous base. The sequence of these bases encodes genetic information. RNA, usually single-stranded, plays diverse roles in protein synthesis and regulation. Understanding the structure and function of nucleic acids is the foundation for all genetic manipulation techniques. Lecture 7, DNA Processes and Basics of a Gene. DNA undergoes 3 key processes: replication, transcription, and translation. Replication ensures genetic information is copied. Transcription converts DNA into messenger RNA, and translation uses ribosomes to assemble proteins. A gene is a specific sequence of DNA that encodes a functional product, usually a protein. Understanding gene structure and regulation is central to genetic engineering and biotechnology applications. Lecture 8, DNA Sequencing Methods. DNA sequencing determines the order of nucleotides in a DNA molecule. Early methods like the Sanger sequencing technique revolutionized genetics, while modern methods use next-generation sequencing, allowing entire genomes to be decoded rapidly. Sequencing is fundamental for identifying genes, diagnosing genetic diseases, and developing personalized medicine. Lecture 9: Fundamentals of Recombinant DNA Technology. Recombinant DNA technology involves joining DNA molecules from different sources to create new genetic combinations. This allows scientists to insert genes into organisms, producing desired traits. Applications include producing insulin through genetically modified bacteria, developing pest-resistant crops, and creating vaccines. Lecture 10: Vector elements and DNA enzymes. Recombinant DNA requires vectors, DNA molecules that carry foreign genes into host cells. Common vectors include plasmids, bacteriophages, cosmids, and artificial chromosomes. Enzymes are crucial tools in DNA manipulation. Restriction enzymes cut DNA at specific sites, ligases join DNA fragments, and polymerases amplify DNA sequences. These molecular scissors and glue are the foundation of genetic engineering. Lecture 11: Constructing recombinant DNA molecules. Constructing recombinant DNA involves isolating the gene of interest, cutting both the gene and the vector with restriction enzymes, inserting the gene into the vector, sealing the DNA fragments with ligase, introducing the recombinant vector into a host cell. The host cell then expresses the inserted gene, producing the desired product or trait. Lecture 12: Plant transformation and gene transfer. Plant transformation introduces new genes into plants to develop improved varieties. Two major approaches exist. Indirect gene transfer, using Agrobacterium tumefaciens, a natural genetic engineer. Direct gene transfer, physically introducing DNA using methods like gene gun, electroporation, or microinjection. Reporter genes and promoters help identify and control the expression of introduced genes. In this first module, we traced the history of biotechnology, explored tissue culture techniques, examined nucleic acids, and introduced recombinant DNA technology. We discussed how genes are sequenced, manipulated, and transferred, particularly in plants. These foundations prepare us for advanced applications in biotechnology.